Chapter 8: Genetic and Molecular Targeted Cancer Drugs and Immunotherapy. Don't miss your chance for synergy.
Both genetic and molecular targeted cancer drugs can work synergistically with immunotherapies to improve outcomes. There are still risks involved, however. In this chapter, I will introduce you to some of the most noteworthy of these drugs. PARP inhibitors. PARP inhibitor, poly-ADP ribose polymerase inhibitor drugs make up a class of medications most typically used with breast and ovarian cancers with BRCA1, BRCA2, or PALB2 mutations. In addition, it then may have some effect in cancers in which the tumor suppressor PTEN gene is defective. PARP is a protein that repairs breaks in the DNA. When the ability to repair these DNA breaks are inhibited, it can lead to the death of the cells. Rapidly dividing cells, such as cancer cells, would be more sensitive to this than normal cells. A study published in the journal Cancer Immunology Research, November 2015, by Higuchi et al., CTLA4 blockage synergizes therapeutically with PARP inhibition in BRCA1 deficient ovarian cancer, demonstrated that CTLA4 inhibitors, but not PD-1, PD-L1 inhibitors, enhance the anti-cancer immune response and survival in the mouse model. However, in a human study with 60 patients with ovarian cancer that was resistant to platinum therapy, 25% of the patients had a complete or partial response using the combination of the PARP inhibitor niraparib with the PD-1 inhibitor pembrolizumab Keytruda. This was versus 5% with the PARP alone and 11% with Keytruda alone. Also, of note, these patients did not have BRCA mutations, which will increase the potential of patients that can be treated with this combination. Studies have shown that PARP inhibitors increase the expression of PD-L1, which can increase the effectiveness of PD-1, PD-L1 inhibitors. Clinical Cancer Research, Zhao et al., February 2017. PARP inhibitor upregulates PD-L1 expression and enhances cancer-associated immunosuppression. Some of the early results with human clinical trials have shown that modest increase in survival time, but overall the results are still fairly lacking. Since we know that in general, for systemic immunotherapy, a PD-1 CTLA4 inhibitor combination, Yervoy Opdivo or Yervoy Keytruda, can be effective at times, those combination of drugs with a PARP inhibitor should be somewhat promising as well. CDK4/6 inhibitors. Cyclin-dependent kinases 4 and 6 inhibitor, palbociclib, Ibrance, is FDA approved for ER-positive and HER2 negative breast cancer. Cyclin-dependent kinases are regulators of cell cycle and cell division and are important for progression of some cancers. In a study published by Goel et al. In Nature, August 2017, they demonstrate that inhibitors of CDK4/6 can promote anti-tumor immunity. One mechanism was by increasing retroviral elements within cancers that resulted in increased double-stranded RNA. This caused an increase in interferons and antigen presentation. In addition, CDK4/6 inhibitors decrease T regulatory cells. These effects result in an increase in the cytotoxic T cell response attacking T cells. Other studies, such as the CDK4/6 inhibitor abemaciclib, induces a T cell inflamed tumor microenvironment and enhances the efficacy of PD-L1 checkpoint blockage by Shah et al., support the idea that a combination of CDK4/6 inhibitors with immunotherapy, such as PD-1, PD-L1 inhibitors should have good synergy. The ability to convert a cold tumor to a hot, inflamed one is a very key area that is lacking in traditional immune checkpoint therapy, leading to failure. Though the human data is still lacking, patients who are getting CDK4/6 inhibitors should certainly look into the options of getting immunotherapy at the same time. EGFR Inhibitors. EGFR inhibitors block the epidermal growth factor receptor, which is found on the surface of some cell-stimulating cell growth. There are several types of EGFR inhibitors. They can be tyrosine kinase inhibitors or monoclonal antibodies. These are used in colon, breast, lung, pancreatic, renal, and head and neck cancers. Although there are very limited studies with the combination of EGFR inhibitors and immunotherapy, theoretically, the combination should be synergistic. However, in limited studies of patients who receive PD-1 inhibitors combined with a TKI EGFR inhibitor, a significant increase in side effects with modest increase in survival was observed. It is unclear what synergy may surface with other immunotherapies in the future. In addition, Schoenfeld et al. Published an article titled, "Severe Immune-Related Adverse Events Are Common with Sequential PD-L1 Blockade and Osimertinib," in Annals of Oncology, March 7, 2019. This article cites a human study which showed that patients first placed on a PD-1 or PD-L1 inhibitor, followed by EGFR inhibitor Osimertinib, Tagrisso, had increased adverse immune side effects. This was greatest if the drugs were started less than 3 months apart. The duration of use of the PD-1, PD-L1 inhibitor did not seem to matter. Of note, if the EGFR inhibitor was used first, no significant immune side effects were seen. Again, systemic use of these drugs, even when not done at the same time, can result in increased side effects. This begs the question, how would systemic EGFR receptor inhibitors work with intratumoral injection of PD-1, PD-L1 inhibitors? Estrogen inhibitors. Estrogen inhibiting drugs are commonly used in estrogen receptor positive breast cancer. These include tamoxifen, fulvestrant, letrozole, and anastrozole. In an article published by Cervonos et al. In the journal Cancer Discovery, January 2017, titled, "Tumor Cell-Independent Estrogen Signaling Drives Disease Progression Through Mobilization of Myeloid-Derived Suppressor Cells," they describe how estrogen inhibits anti-cancer immunity irrespective of the receptor status of the cancer. It is known that there are inherent differences in the immune systems of men and women, particularly related to hormonal differences. Estrogen receptors are not only expressed on some tumors, but are also expressed on immune cells. Studies are indicating that estrogen causes an increase in regulatory cells, such as MDSCs, along with reducing infiltration of CD8 positive, tumor-attacking T cells in the tumor microenvironment. The basic idea is that estrogen-inhibiting drugs seem to have positive effects on the anti-cancer immune response, no matter if the cancer cells are estrogen receptor positive or not. VEGF inhibitors. In February 2019, Motzer et al. Published in the New England Journal of Medicine results of a human study with the combination of avelumab, PD-L1 inhibitor, with axitinib, and LIDA, a TKI inhibiting VEGF, c-KIT, and PDGFR. This study showed that the combination extended progression-free survival by an additional 5.4 months over standard of care Sanitinib 8.4 months to 13.8 months. I know this is modest, and I would certainly hope for better, but since axitinib is a pill, it may be easy to add for patients who are having to stick more to standard of care immunotherapy. One of the classic VEGF inhibitors is the monoclonal antibody drug bevacizumab, Avastin. In the clinical trial called iEMPOWER 150, they studied 1,200 advanced non-small cell lung cancer patients. The results showed that at 1 year, the survival of the combination bevacizumab with the PD-L1 inhibitor atezolizumab Tecentriq was 37% versus 18% for bevacizumab plus chemotherapy. In addition, they noted that patients with gene mutation had a similar response, when normally it was thought those patients did not have much benefit from immunotherapy. VEGF is mainly known for its effect on angiogenesis, enhancing blood vessel growth to tumors. However, VEGF also causes immunosuppression in the tumor microenvironment. One aspect, as mentioned before, is that immune cells do not just swim to the tumor, they must be attracted to the area by the cells lining blood vessel walls. Excess VEGF in the tumor microenvironment leads to vessel formation that impedes the ability of immune cells to enter. Essentially, it helps create a barrier to the immune system. In addition, VEGF causes the increase of regulatory tumor-protecting immune cells such as MDSCs and TREGs. Also, VEGF inhibits the maturation of dendritic cells. Mature dendritic cells present antigens to the other immune cells and stimulate an immune response, while immature dendritic cells function as regulatory cells inhibiting an immune response. For all of these reasons, it makes sense that anti-VEGF drugs not only are effective against tumor vessel development, but also are actually immunotherapy agents in their own right. There are numerous anti-VEGF drugs that have FDA approval besides the ones mentioned. Others currently in trials combined with immunotherapy include ramucirumab, Cyramza, and nintedanib, vargetex-ofev. CXCR4 inhibitor Moravovac. Different than some of the other agents listed in this chapter, this is not only a targeted agent, but is also an off-label medication as well. Moravovac is actually an FDA-approved antiviral agent used for the treatment of HIV. This medication received FDA approval back in August 2007. Besides allergic and skin reactions, this drug does have liver toxicity, so liver functions need to be closely monitored. An article published by Aldinici and Casagrande in the journal International Journal of Molecular Sciences, May 2018, titled Inhibition of CXCL5 Axis Against the Progression of Gastric Cancer, gives a great explanation of the underlying mechanism. To give you a brief explanation, CXCL5 binds to CXCR4, causing the activation of the receptor. The activated receptor has multiple mechanisms of cancer stimulation. First, it causes direct proliferation of the cancer through mTOR, cyclin D1, c-MYC, and DAD1, along with increasing glucose metabolism. Second, it causes direct immunosuppression by recruiting regulatory immune cells, especially TAMs, that ultimately lead to the increase in inhibitory cytokines such as IDO, TGFB, and IL-10. Halama et al. Demonstrated that blocking CXCR4 with Moravovac led to converting tumor-associated macrophages, TAMs, from the M2 tumor-protecting to the M1 tumor-attacking type. Besides TAMs, it also seems that Moravovac reprograms MDSCs as well. If that was not enough, Moravovac also inhibits VEGF, tumor migration, and metastasis formation. MEK inhibitors. In 2016, results were presented at the European Society for Medical Oncology, ESMO, by Johanna Bendel, MD, on phase 1 study combining atezolizumab, PD-L1 inhibitor, with cobimetinib, MEK inhibitor, for patients with microsatellite stable MSS metastatic colorectal cancer. In general, MSS patients do not respond as well to immunotherapy and unfortunately make up the majority of patients, 95%. This combination offers the hope that MSS metastatic colorectal patients may have an improved chance to respond to one of the most widely used type of immunotherapy, PD-1, PD-L1 inhibitors. The results were pretty modest, with 17% partial response, 22% stable disease, with a response duration from 4 months to greater than 15 months. The exact mechanism still seems to not be well understood, but most likely involves a decrease in immune suppressive mediators such as COX2 and ARG1 with increasing cytotoxic T-cell infiltration in the tumor microenvironment. Ebert et al. Published a study in Immunity 2016 that MEK inhibition reduces CD8+ T-cell apoptosis during chronic T-cell receptor stimulation. Often when the immune system is chronically activated, like in cancer, the immune cell that attacks the cancer can be fatigued and may die off. This indicates an MEK inhibitor may assist in preventing this immune suppression from developing. Certainly, in patients who have MSS colorectal cancer and are considering systemic immunotherapy with a PD-1, PD-L1 inhibitor, this combination with MEK inhibitor should be an option. ALK inhibitors. Combinations can also be associated with problems. In an article published by Patel et al. In Journal of Thoracic Disease, July 2018, titled LAK Inhibitors and Checkpoint Blockade, a cautionary tale mixing oil with water, they describe the high incidence of hepatotoxicity, liver toxicity, with this combination. In this study, there were deaths due to the hepatotoxicity, leading to the trial being discontinued. There were a few things of note here. Even though PD-1, PD-L1 inhibitors have been considered a major advancement, they still have an overall low success rate and mild toxicity. In the future, I suspect this class of drugs will be replaced or used as a follow-up to other immunotherapies. In addition, toxicity from combinations that could be highly effective may be avoided by intratumoral injection. CXCR4 inhibitors plerixafor. Cancer cells can have increased expression of CXCR4, which binds to CXCL12. This interaction can function as a homing mechanism for cancer. It also can increase fibrosis in the cancer tissues, which acts as a barrier from immune cells. In breast and lung cancer, patients with overexpression of CXCR4 have a worse prognosis. CXCR4 is also involved in other cancers such as prostate, ovarian, colon, and bladder. The drug plerixafor is FDA approved for the treatment of lymphoma and multiple myeloma. This allows for the off-label use in other cancers. In a study published in PNAS, January 30th, 2019, by Chen et al., titled Blocking CXCR4 Alleviates Desmoplasia, Increases T lymphocyte Infiltration, and Improves Immunotherapy in Metastatic Breast Cancer, they describe how plerixafor enhances immune checkpoint therapy in mouse model. This could lead to another target in immunotherapy. It is certainly possible to evaluate the patient's tumor for expression of CXCR4 to see if they may have potential benefit. DDR2 inhibitors. The most potent FDA-approved DDR2 inhibitor is the Sanitinib, Sprycel, which is used in CML and ALL. It is a tyrosine kinase inhibitor, which besides DDR2 also targets BCR, ABL, SRC, c-KIT, and ephrin receptors. In a study published in Science Advances February 20th, 2019, by Tu et al., titled Targeting DDR2 Enhances Tumor Response to Anti-PD-1 Immunotherapy, they described the mechanisms of synergy in mouse model. They described that there is an increased infiltration of CD8 positive T cells in both the tumor and spleen. This was unique to the combination. Though this is a limited study and human studies are needed, it does show promise that the Sanitinib may enhance the effectiveness of PD-1 inhibitors, the most widely used type of immunotherapy at this time. Still, there's more that can be done to empower the immune system to do its job, and this is where the patients themselves come in. There are some simple but novel steps that cancer patients can take to enhance the immune response. It's not just a gut feeling that I have that tells me cancer can be cured, it's the gut itself. Because believe it or not, what grows inside our gut can help our immune system save our lives. So turn the page and find out more.